Pain is not a direct readout of tissue damage. It is a constructed experience that depends heavily on how signals are processed and filtered within the nervous system. One of the most important control points in this process is the spinal cord, where incoming pain signals from the body first arrive before being transmitted to the brain. At this level, specialized circuits act as a gate, determining how much of that signal is allowed to pass through. This concept is often referred to as neural gating, and it provides a powerful mechanism through which the brain can regulate pain in real time. When nociceptors in the body detect potentially harmful stimuli, they send electrical signals along peripheral nerves into the dorsal horn of the spinal cord. Here, these signals encounter interneurons that can either amplify or inhibit the transmission. The state of these interneurons is not fixed. It is influenced by both local inputs and descending signals from the brain. This means that the brain is constantly sending instructions down the spinal cord, adjusting how incoming sensory information should be interpreted. One of the key pathways involved in this top-down control originates in the periaqueductal gray, a region located in the midbrain. This structure receives input from higher brain areas involved in emotion, cognition, and expectation, such as the prefrontal cortex and amygdala. When activated, the periaqueductal gray sends signals to another region called the rostroventral medulla, which then projects down to the spinal cord. These descending pathways release neurotransmitters, including endogenous opioids like endorphins and enkephalins, which act to inhibit pain transmission at the synaptic level. These endogenous opioids bind to receptors on neurons in the spinal cord, reducing the release of excitatory neurotransmitters and decreasing the likelihood that pain signals will be passed upward to the brain. In effect, they turn down the volume of the signal before it ever becomes a conscious experience. This system is highly adaptive and can be engaged in various situations. For example, during intense physical activity or in high-stress environments, the brain can suppress pain to allow continued performance, a phenomenon often observed in athletes or during emergencies. The gating system is also influenced by non-painful sensory input. Signals from touch receptors, for instance, can compete with and suppress pain signals at the spinal level. This is why rubbing or applying pressure to an injured area can reduce the sensation of pain. These competing inputs activate inhibitory interneurons that reduce the transmission of nociceptive signals, effectively closing the gate. Importantly, this system is not only reactive but can be shaped over time through experience and behavior. Repeated exposure to manageable levels of discomfort can enhance the efficiency of descending inhibitory pathways, increasing the brain's ability to regulate pain. Conversely, chronic stress, anxiety, or persistent focus on pain can weaken these pathways, leading to heightened sensitivity and amplification of pain signals. The flexibility of this gating mechanism highlights that pain is not solely determined by physical input, but by how the nervous system chooses to process that input. By influencing the brain regions that control descending pathways through attention, mindset, and controlled exposure to stress, it is possible to alter the intensity of pain at a very fundamental level within the nervous system. Attention acts as a powerful control system that can increase or decrease the intensity of both pain and pleasure by directly modulating activity in specific brain circuits. Sensory input does not arrive in the brain as a fixed objective signal. Instead, it is shaped by where your focus is directed. When you attend closely to a sensation, neurons representing that sensation increase their firing rates, and more cortical resources are allocated to processing it. This amplification occurs in regions such as the somatosensory cortex and the anterior cingulate cortex, both of which are heavily involved in the perception of pain. When attention is narrowly focused on a painful stimulus, the brain effectively increases the gain on that signal. Functional imaging studies show that directing attention toward pain increases activation in pain-related areas, making the experience more intense and more salient. This is why minor discomfort can become overwhelming if you fixate on it. The brain interprets the focused attention as a sign that the stimulus is important and potentially threatening, and it adjusts perception accordingly. On the other hand, shifting attention away from pain can significantly reduce its perceived intensity. This is not just distraction in a casual sense. It involves actively engaging neural circuits that compete with pain processing. When attention is directed toward a cognitively demanding task, a meaningful goal, or even a rewarding experience, the brain reallocates resources. Prefrontal cortical areas become more active, exerting top-down control that suppresses activity in pain processing regions. This reduces both the sensory and emotional components of pain. This mechanism also explains why pain can feel less intense during activities that require full engagement, such as sports, creative work, or problem solving. In these states, attentional networks are heavily occupied, leaving fewer resources available to process pain signals. The same principle applies to pleasure. When attention is fully directed toward a positive experience, the brain enhances the activity of reward-related circuits, including those involving dopamine and endogenous opioids. This increases the depth and richness of the pleasurable experience. Attention is closely linked to expectation and interpretation. If you expect a sensation to be painful and direct your focus toward it, the brain prepares for and amplifies that experience. This involves interactions between the prefrontal cortex, which generates predictions, and subcortical structures that process sensory input. In contrast, if attention is directed in a way that reinterprets the sensation, viewing it as neutral or even beneficial, the same physical input can feel less aversive. Training attention can therefore change how pain and pleasure are experienced over time. Practices like mindfulness meditation are particularly effective because they teach the ability to observe sensations without automatically reacting to them. This changes the relationship between attention and sensation, reducing the tendency to amplify discomfort. Instead of narrowing focus on the most intense aspect of the experience, attention becomes broader and more stable, which decreases the overall impact of pain signals. There's also a temporal dimension to attention. Rapid shifts in focus can prevent the buildup of sustained neural activity associated with pain. By periodically redirecting attention, the brain does not allow pain-related circuits to dominate for extended periods. This dynamic control over focus creates a more flexible and adaptive sensory experience, where neither pain nor pleasure becomes overwhelming or uncontrollable. The experience of pleasure is governed by an interaction between dopamine and endogenous opioid systems, and understanding how these two systems work together provides a direct way to influence motivation, satisfaction, and even the perception of pain. Dopamine is primarily associated with pursuit, anticipation, and the drive to obtain something rewarding. It is released in brain regions such as the ventral tegmental area and projected to the nucleus accumbens and prefrontal cortex. When dopamine levels rise, it increases focus, energy, and the willingness to exert effort. This is the system that makes you want something, not necessarily the one that makes you enjoy it. Endogenous opioids, including endorphins and enkephalins, are more closely tied to the actual experience of pleasure or relief. These molecules are released during rewarding experiences and bind to opioid receptors in the brain, producing feelings of satisfaction, calm, and reduced pain. While dopamine drives the behavior toward a goal, opioids provide the sense of completion once the goal is achieved. This distinction is critical because it explains why chasing rewards and experiencing them can feel very different at a neural level. The timing of these systems matters. Dopamine tends to peak before or during the anticipation of a reward, especially when there is uncertainty or variability. This is why unpredictable rewards, like social media notifications or intermittent success, can be highly engaging. Once the reward is obtained, dopamine levels often drop, and endogenous opioids take over to generate the feeling of pleasure. If rewards are too frequent or too intense, this system can become dysregulated. The brain may reduce dopamine receptor sensitivity or baseline dopamine levels, leading to a state where more stimulation is required to achieve the same level of motivation or excitement. This dynamic has direct implications for both pleasure and pain. When dopamine levels are chronically elevated through constant stimulation, such as excessive sugar intake, digital consumption, or other high-reward behaviors, the baseline level of motivation can decrease over time. This creates a contrast effect where ordinary activities feel less rewarding and more effortful. In parallel, the opioid system can also become less responsive, reducing the sense of satisfaction derived from experiences that would normally be pleasurable. On the other hand, spacing out rewarding experiences and engaging in effort-based activities can restore sensitivity in these systems. When dopamine is allowed to return to baseline between peaks, the system becomes more responsive. Activities that require effort, such as exercise or skill learning, tend to produce a more balanced release of dopamine and endogenous opioids. This not only enhances motivation, but also leads to a more sustained and stable sense of pleasure. There's also an interaction between these systems and pain perception. Endogenous opioids released during rewarding or meaningful activities can reduce the intensity of pain by acting on the same receptors targeted by pain relief medications. This means that increasing natural opioid activity through positive experiences can simultaneously enhance pleasure and decrease discomfort. The balance between dopamine-driven pursuit and opioid-driven satisfaction is not fixed. It is shaped by behavior and environment. By managing the frequency, intensity, and context of rewarding stimuli, it is possible to recalibrate these systems, leading to greater control over both motivation and the quality of subjective experience. Pain and pleasure are tightly linked through what is known as the opponent process system, a fundamental principle in neuroscience that explains how the brain maintains balance. Whenever you experience a strong deviation in one direction, either intense pleasure or significant discomfort, the nervous system initiates a compensatory response in the opposite direction. This is not optional. It is built into the way neural circuits regulate stability, or what is called homeostasis. When you engage in a highly pleasurable activity, such as consuming sugar, achieving a goal, or experiencing novelty, there is a surge in dopamine and endogenous opioids. This creates a temporary state of elevated mood and motivation. However, shortly after this peak, the brain activates counteracting mechanisms to bring activity back down to baseline. This often results in a dip below baseline, where you may feel less motivated, slightly irritable, or even fatigued. The more intense and frequent the pleasure stimulus, the stronger this rebound effect tends to be. Over time, repeated exposure to high levels of stimulation can deepen these dips, making baseline states feel less satisfying. The same process works in reverse for pain or discomfort. When you undergo a challenging or stressful experience, such as intense exercise, cold exposure, or effortful mental work, the initial state is characterized by discomfort and strain. During this phase, stress-related systems are activated, including the release of cortisol and increased neural activity in areas associated with effort and pain. But once the stimulus is removed or completed, the brain initiates a rebound response that often leads to a rise in mood, relaxation, and even a sense of pleasure. This is partly driven by increased release of endogenous opioids and a recalibration of dopamine signaling. A key structure involved in tracking these shifts is the habenula, a small region of the brain that monitors negative outcomes and modulates dopamine activity. When experiences are worse than expected, the habenula suppresses dopamine release, reinforcing avoidance. When the brain detects relief from discomfort or a positive contrast, dopamine activity can increase, enhancing the sense of reward. This dynamic contributes to the way the brain learns from both pain and pleasure, adjusting future behavior based on past outcomes. This opponent process has important implications for how behavior shapes long-term emotional states. If someone consistently seeks out high-intensity pleasure without allowing for recovery or baseline restoration, the system becomes skewed. The brain spends more time in the down phase, leading to reduced motivation and increased reliance on stronger stimuli to feel normal. In contrast, deliberately introducing manageable levels of discomfort can strengthen the system's resilience. The rebound from these experiences can elevate baseline mood and increase sensitivity to everyday rewards. Timing and repetition are critical factors. Occasional exposure to discomfort can produce noticeable positive after effects, but consistent, controlled exposure tends to produce more lasting adaptations. The brain begins to anticipate not just the discomfort, but the reward that follows, which can shift perception and reduce the aversiveness of the initial experience. This is one reason why trained individuals often report less discomfort during challenging activities. They have effectively reshaped the balance between the opposing processes. The system is constantly recalibrating based on recent experiences. By understanding that every spike in pleasure carries a cost and every bout of discomfort carries potential benefit, it becomes possible to structure behavior in a way that stabilizes mood, enhances motivation, and reduces the extremes of both pain and pleasure. Breathing is one of the most direct and accessible ways to influence the nervous system, and it has a measurable impact on how pain and discomfort are perceived. Unlike many physiological processes, breathing sits at the intersection of conscious and unconscious control, meaning you can deliberately change it and immediately alter your internal state. This influence occurs through its effects on the autonomic nervous system, which regulates arousal, stress, and recovery. When you engage in slow, controlled breathing, especially with an emphasis on longer exhales, you activate the parasympathetic branch of the autonomic nervous system. This branch is associated with rest, recovery, and reduced physiological arousal. The vagus nerve plays a central role here, carrying signals from the brain to the heart, lungs, and other organs. Slow breathing increases vagal tone, which in turn lowers heart rate, reduces blood pressure, and decreases the release of stress hormones like cortisol. These changes create a physiological environment that dampens the intensity of pain signals and reduces the emotional reactivity associated with discomfort. At the level of the brain, controlled breathing influences activity in regions involved in both pain processing and emotional regulation, including the insula, anterior cingulate cortex, and prefrontal cortex. By stabilizing neural activity in these areas, breathing can reduce the amplification of pain that often occurs when stress and anxiety are high. In this way, breath control does not eliminate the source of pain, but changes how the brain interprets and responds to it. The rhythm of breathing also matters. Slow breathing, typically in the range of about 5 to 6 breaths per minute, has been shown to synchronize physiological systems, including heart rate variability. Higher heart rate variability is associated with greater adaptability and resilience, and it correlates with a reduced perception of pain. This synchronization effect creates a more balanced internal state, making it easier for the brain to regulate incoming sensory information without overreacting. In contrast, rapid and shallow breathing tends to activate the sympathetic nervous system, which is responsible for the fight-or-flight response. This state increases alertness and can be useful in situations that require immediate action, but it also heightens sensitivity to pain and stress. Elevated breathing rates can lead to increased carbon dioxide loss, which can alter blood pH and contribute to sensations of anxiety or discomfort. This physiological shift can amplify the perception of pain, making it feel more intense and harder to manage. There is also a cognitive component to breathing. Focusing on the breath provides a stable anchor for attention, which can reduce the tendency to fixate on pain. This dual effect, physiological regulation combined with attentional control, makes breathing a powerful tool for modulating both the sensory and emotional dimensions of discomfort. Over time, consistent practice can strengthen the connection between breath control and nervous system regulation, making it easier to access this state even under stress. Different breathing patterns can be used strategically depending on the goal. Slow, deep breathing is effective for reducing pain and promoting relaxation, while more deliberate patterns, such as brief breath holds or cyclic breathing, can build tolerance to discomfort and improve resilience. The key point is that by changing how you breathe, you are directly influencing the systems that determine how pain and pleasure are experienced. At both the expectation and learning play a central role in shaping how pain and pleasure are experienced. And this process is rooted in the brain's ability to continuously predict and update outcomes based on past experience. The brain is not simply reacting to sensory input in real time, it is constantly generating predictions about what is likely to happen next. These predictions influence how incoming signals are interpreted, often before they even reach conscious awareness. This predictive system involves close interaction between the prefrontal cortex, which generates expectations, and deeper brain structures that process sensory and emotional information. One of the clearest demonstrations of this process is the placebo effect. When a person believes that a treatment will reduce pain or enhance pleasure, that belief alone can trigger measurable biological changes. The brain releases endogenous opioids and dopamine, which can reduce pain perception and increase feelings of well-being. This is not imagined or superficial. It involves the same neural pathways that are activated by actual pharmacological interventions. The expectation of relief or reward essentially primes the nervous system to produce that outcome. The opposite effect, often referred to as the nocebo response, also highlights the power of expectation. If a person anticipates pain or negative outcomes, the brain can amplify discomfort by increasing activity in pain-related circuits and reducing the effectiveness of natural pain-inhibiting systems. This demonstrates that expectation does not just color experience, it can directly increase or decrease the intensity of physical sensations. Learning reinforces these effects over time through a process of neuroplasticity. Each experience of pain or pleasure strengthens specific neural pathways, making those responses more likely to occur in the future. If a particular stimulus is repeatedly associated with pain, the brain becomes more efficient at producing that response, sometimes even in the absence of the original trigger. Similarly, if certain behaviors or environments are consistently linked to positive outcomes, the brain enhances its sensitivity to those cues, increasing motivation and anticipation. This learning process involves multiple brain regions, including the hippocampus, which encodes contextual memory, and the amygdala, which assigns emotional significance to experiences. Together, these structures help form associations between specific situations and expected outcomes. Over time, these associations can become automatic, shaping behavior and perception without conscious effort. For example, if someone repeatedly experiences relief after a certain action, the brain begins to anticipate that relief, reducing pain even before the action is completed. Importantly, these learned patterns are not fixed. Because the brain remains plastic throughout life, expectations and associations can be updated with new experiences. Deliberately changing how you interpret and respond to sensations can gradually reshape these neural circuits. This might involve reframing discomfort as a signal of growth or progress, or intentionally focusing on positive aspects of an experience to strengthen rewarding associations. The consistency of these new experiences is critical. Occasional changes in perception may not be enough to override deeply ingrained patterns, but repeated exposure to new interpretations can shift the brain's default responses. Over time, the predictive system becomes recalibrated, leading to a different baseline for how pain and pleasure are processed. This means that by consciously shaping expectations and reinforcing new learning patterns, it is possible to influence the underlying biology that determines subjective experience. When you look across all these mechanisms together, it becomes clear that pain and pleasure are not fixed outputs, but dynamic processes shaped by attention, expectation, neurochemistry, and learned experience. The brain is constantly adjusting its responses based on what you repeatedly do, think, and feel. By deliberately training attention, regulating breathing, managing reward exposure, and reshaping expectations through consistent behavior, you can gradually recalibrate these systems. Over time, this leads to a more balanced baseline where pain becomes more manageable, pleasure becomes more sustainable, and your overall sense of control over internal states becomes significantly stronger.